Okay, my name is Carina Collard. I'm a senior environmental biology and climate change major, and I'm minoring in environmental science and policy as well. Today we'll be talking about my honors thesis, which was funded by the Hook Fellowship and is on the effects of urbanization on riparian vegetation community structure. So, as we all know, the global population has undergone a near exponential increase in the past 200 or so years. In 1800, the global population was about 1 billion, but it's increased by more than 7 billion people in that time span. And at the same time, we've seen an increase of people moving from rural areas to urban areas. In 1800, less than 10% of the global population lived in urban areas, but by 2050, that number is expected to exceed 65%. And Austin, Texas is no exception to this trend. In fact, every single year from 2010 to 2020, Austin was the fastest growing metropolitan area in the United States. So we gained about 1 million people in our population, which is a 33% increase in a single decade, which is massive. So that shift of human populations from rural to urban communities is known as urbanization. And there are a lot of very well documented negative consequences associated with this process, and that's because it induces additional anthropogenic stressors on ecosystems. Anthropogenic stressors are human-caused stressors that tend to diminish an ecosystem. So that can result in changes in biogeochemical processes, habitat fragmentation, loss of biodiversity, increase of invasive species, and alteration of land use change. Altogether, this will degrade ecosystems and will diminish or completely eliminate their ability to provide ecosystem services. It's also important to note that the effects of urbanization are not a monolith. So as you move closer to more densely populated areas of the city, these effects tend to be more pronounced, whereas you move closer to the suburbs where there's less dense human population, the effects will be less severe. So this term that I mentioned on the last slide, ecosystem services, refers to the indirect and direct contributions of ecosystems to human well-being. In ecology, we divide ecosystem services into 4 main categories: supporting, provisioning, regulating, and cultural. Some of these services are very tangible, very direct, like timber or meat from an ecosystem. Some of these services are less tangible and more kind of indirect, like the recreational or aesthetic value of a beautiful ecosystem. But regardless, all of these ecosystems are invaluable to human well-being and life on Earth. In fact, these ecosystem services are incredibly important, especially during a time where the world is getting more urbanized and the population is increasing, because ecosystems with a high potential to provide ecosystem services are one of the best ways to mitigate the negative consequences associated with urbanization. One such ecosystem with a very high potential to provide ecosystem services are riparian areas, and those are the lands between terrestrial and aquatic systems. They're these kind of transitional zones of energy and material exchange that provide a disproportionate number of ecosystem services for the amount of space that they take up. So they do things for us like mitigate floods, they sequester CO2, they filter water, they provide erosion control, and they're habitats that support very high biodiversity. And many of these services that riparian areas provide are due to the density and diversity of plant species there. So basically, how many different species of plants and what species of plants are there. So it's safe to say that the functioning of these riparian systems is directly related to their vegetation structure and composition. This figure over here shows kind of an ideal riparian area. As you see, it has an understory of like herbaceous things, the sedges and the rudges, but then it also has an overstory of woody plants and trees. So an intact riparian system would have both a healthy understory and a healthy overstory. And all of these services that I mentioned on the last slide basically contribute directly to the health and functioning of freshwater ecosystems. So our river health, our stream health, everything is dependent on having intact riparian vegetation communities. But unfortunately, these ecosystems are highly vulnerable because they're subject to anthropogenic stressors from both terrestrial and aquatic ecosystems. That can lead to alteration of hydrology, invasive introduction, land use change, and eutrophication. My study specifically focused on the introduction of invasive species. This is a huge problem because it can completely change the structure and composition of these vegetation communities. This picture on the top over here is a picture of Blund Creek Preserve. As you can see, the trees in that area are very small and they're very clustered together, and there's no grass, it's just bare ground. Those trees are called privet, and it's a very invasive species. The picture below is of Emma Long Park, and as you can see, there's a very healthy understory with lots of different grasses, and then there's only a few large trees, and those are all actually native species. So that's just a good example of how invasives can completely change the structure of an ecosystem. So despite the importance of this ecosystem and despite the changes that we're seeing in the world, there has been inadequate research conducted thus far to aid in their conservation, specifically looking at changes in plant community structure associated with urbanization and kind of broader land use change have been overlooked in place of studying ecophysiological measurements specifically. Therefore, the objective of my study was to identify patterns of riparian plant vegetation structure and community composition across an anthropogenic gradient. And the broader goal of this study was to create knowledge that would help aid in the development of adaptive management strategies and research-based public policies to conserve these invaluable ecosystems. So to address the knowledge gaps, I posed the question of how the degree of urbanization would influence riparian vegetation community composition and structure. I hypothesized that more urbanized habitats would have a different forest structure with smaller, younger trees and less understory diversity. I had a few more specific predictions about dependent variables, but for the sake of time, I'm going to skip past those, but I will take questions about that later. So my study area was the greater Austin, Texas metropolitan area, including the Balcones Canyon Land Preserve. As I mentioned, this project was funded by the Hook Fellowship from Wild Basin, which is part of the BCP, so I chose a few other sites within the BCP as well. And Austin is really an ideal city for a case study like this because, as I mentioned at the start of the presentation, it's very rapidly urbanizing and rapidly growing, but at the same time, the city has managed to preserve more than 2,000 acres of land, so it's a great place to kind of compare things in preserved and unpreserved areas. So for site selection, I chose 8 different sites. 4 sites I grouped into the land use category of suburban or urban, so they kind of are probably more subject to anthropogenic influences. Those sites are marked by a blue dot, and then 4 other sites I grouped into a land use category of preserve and ex-urban, which were marked by green dots. And at each site, I had a specific selection criteria for where I was collecting data. I wanted areas with natural substrate that were fairly easy to access. I wanted a minimal curve along the bank and then a minimal change in elevation from the water to the bank. So for data collection, I set up a 25 meter by 5 meter fixed area quadrant to assess the overstory. Within that quadrant, I was looking at trees taller than a meter in height. I identified them to the species level using a combination of iNaturalist and field guides, and then I measured their diameter at breast height, also known as DBH, which is basically exactly what it sounds like. It's the diameter of the tree at approximately your breast height. And then within my larger fixed area quadrant, I set up 6 2 meter by 2 meter nested plots to assess the understory. For the understory, I was looking at forbs, herbaceous species, and trees less than a meter in height. I identified them again to the species level. For the understory plants, I estimated the amount of ground that they were covering, and for the trees, I again measured their DBH. All of my data analysis and visualization was done using RStudio version 4.32. I ran independent t-tests to compare my dependent variables across the land use groupings of preserve, ex-urban, and suburban urban. And then I ran simple linear regressions to determine if the proportion of invasive trees was a useful predictor of my dependent variables. My dependent variables are off in this table to the side. I already explained DBH. I also had total basal area, which is basically the combined DBH of all the trees in a plot. And then I had the amount of vegetated ground cover, the stem density, which is the number of trees per plot, the species diversity, and then the proportion of invasive individuals. So the results for my t-tests yielded no statistically significant differences between groups. As you can see, I had p-values all quite a bit above 0.05. However, there were a few observable differences between the groups for some variables. On the left, I have a bar graph that shows average DBH on the y-axis and land use group on the x-axis. Preserve and ex-urban is green, and suburban and urban is blue. So what this tells us is that in preserve sites, the trees did have a larger DBH than the urban-suburban sites, though it wasn't statistically significant. The figure on the right shows the average proportion of invasive trees on the y-axis and land use on the x again. And that shows that there was fewer invasive trees in preserved areas than there were in urban areas. So that really aligns with those pictures I showed you at the beginning. Areas with more invasive trees have a smaller DBH, and bigger native trees, bigger DBH. So for the linear regressions, I found one statistically significant relationship, which was between the average ground cover and the amount of invasive species. And I had a p-value of 0.52 with some other observable relationships. The figure right here, total basal area on the y-axis, proportion of invasive trees on the x, not statistically significant. I had a p-value of 0.23, but as you can see, there's sort of a correlation maybe between increased total basal area and the proportion of invasive trees. My statistically significant finding is this figure over here. It's average vegetated cover on the y, proportion of invasives on the x, and as you can see, the amount of vegetated ground cover greatly decreases with the amount of invasive species, which also aligns with those pictures I showed at the beginning. Lots of invasive trees, no grass on the ground. So the conclusions that I got from this study basically were that my results suggest that the land use groupings of urban and suburban and preserve and exurban were not the most meaningful ways to kind of explain the differences in these riparian vegetation communities. And also that the proportion of invasives was not the most meaningful metric of urbanization. However, it was stronger than grouping them into these land use categories. My kind of hypothesis for why I got these results is that the differences between the sites that I chose were just too great. So it turns out, once I collected all my data, I found that each site was dominated by a different tree species. Some were dominated by privet, some were dominated by ash juniper or bald cypress. So being dominated by a different species can, like I said, really change the structure in a way that wasn't accounted for in my statistical analyses. And also the sites that I chose spanned multiple waterways. Watersheds are kind of a big way that invasives get spread. If seeds travel, they can end up on the bank. So the fact that these sites were in different watersheds, again, introduced some variation that I wasn't able to account for in my statistics this time around. So future directions for this study, of course, add additional sites, and I would probably want to do 2 to 3 replicates of a quadruped at each site. And I would also be sure to choose sites with a shared dominant species and probably a shared watershed as well. I want to give a few acknowledgments to the Hook Fellowship for funding this project. My advisor, Dr. Concilio, Dr. Eric Johnson helped me a lot with experimental design. My lab group provided a lot of feedback for me, and my girlfriend helped me a lot with my data collection. So I wanted to thank all of those people. And now I will take questions.
So we have plenty of time for questions.
Yes.
So this is going to sound weird. I know, okay, obviously urbanization has some negative consequences. Are there any positives to urbanization? I'm thinking of as population increases, is it better to like stick us all in one spot? And then down to more open spaces in the rest of the world? You know, otherwise we're just spread out everywhere and we corrupt everything.
I think generally speaking, we kind of were talking about this actually at the climate justice teach-in the other day. We were saying that it's better to kind of build cities in a more dense way because that kind of yields a city to be more walkable, and transportation is like a huge thing that leads to negative consequences associated with urbanization. So I think to a degree that could be true. Also, we usually find that urbanization will increase species diversity, but that increase in diversity is because it's more invasive species. So it's like, yeah, there's higher biodiversity, but it's because there's more introduced invasives.
Gotcha. Does the city of Austin do anything to mitigate invasive species in their parks department? I mean, is there already any policies related to this?
So in the preserved areas, they all will have their own management plans, which usually will involve invasive species removal. But the issue with conservation all around is that there's not enough resources, there's not enough money or people to do the work. And pulling out invasives is almost lost work at a point because you have to pull up the whole root system for some things, and it's not really feasible to do. So there are management strategies in place for different places, which is another thing that could have contributed to differences observed. There's also different policies in place regarding the amount of impervious cover, so the amount of like roads and pavement that can be built in East and West Austin. In West Austin, there is a 25% limit on impervious cover, but in East Austin, there's no limit on impervious cover. You can just pave everything you want. So that can also create differences. Some of my sites that were in urban or suburban areas were in the east side and some were in the west side, so that's more differences. Other questions?
Did you notice more invasive species on the east side?
Yes. Yeah. Definitely more privet and like smaller diameter herbaceous stuff.
curious, so what are your future directions? Is to sample from within a watershed?
Mm-hmm.
Is that possible to, I don't know much about this. Are there watersheds that are big enough to get both like a suburban and a preserve sample within the same watershed?
I can't definitively answer that, but I think yes. I think so. Like some of the places, or a few of them that I went to were in Boggy Creek, and some of the sites on Boggy Creek are like in East Austin, but some are kind of less, like in more suburban areas. So I think that would be possible. Yeah, that could be very interesting.
Any other questions? Let's thank Tina and this time I would invite her mentor to come up, but unfortunately Dr. Concealio couldn't be here today, but Dr. Sasha West is going to read some work that Dr. Concealio did. And yes, and she'll also present the liquor consultant report.
Okay, I'm going to do this part
She will
first.
be able to wear at graduation to represent as a honor student.
Congratulations. Thank you. Well, I will say Amy loves you and your work so much that this is what she sent me. But I've been instructed to shorten it because we're in the last session of the day, and then I'll send this home with you. Amy just was so impressed with the research that you did. I know that she took over for Kim O'Keefe on this work with the Hook Fellowship. But what she says is, in addition to having her in my classes and working with her on campus and environmental initiatives, I knew Nina well from lab meetings and field outings that Kim and I jointly hosted over the last few years. Nina got my guidance on a couple of things as she was refining the methods, and we brainstormed about how to analyze her results, but she did the bulk of the work on her own. She conceived the study design, carried out the field work, ran the analyses, and put together the presentation that she practiced in our lab meeting a few weeks ago. I am so impressed with what she accomplished over this past year, and I would say that too in poetry, which is a completely different field. And it has been an absolute pleasure working with Nina on her honors thesis and everything else. I'm really going to miss having her around campus when she graduates. I have a feeling a lot of us would say that. Roy, would you say that? Yeah, I think that's pretty common. And I hope she stays in touch with us and keeps coming back for visits to share what she's up to, which will no doubt be inspiring, interesting, and important work. Congratulations, this is really wonderful.
Thank you.
And bravo, yay.